Hello, everyone. Welcome to today's SONA and Trend in Africa webinar series. My name is Mahmoud Bukarmena, and I'm a research fellow in Sussex Neuroscience and also a member of SONA and Trend. I'm one of the organizers of this webinar series, and the webinar series is in partnership with African Academy of Neurology, UK Dementia Research Institute, International Parkinson's Disease Genomics Consortium, Africa, and we are part of Worldwide Neuro. And it's really exciting to have Professor Karen Duff with us today. And I have to give the whole credit to my colleague, Mosab, who invited her to give this talk today. So Mosab, thank you. I'm going to tell you a little bit about Karen, and hopefully she will say more about herself. So Professor Karen Duff has worked on AD and the tauopathies for more than 25 years and received the Potemkin Prize for this work in 2006. She has a strong interest in neurodegenerative diseases etiology and has created various mouse models for AD, tauopathy, and synaptopathy, which have facilitated the study of many aspects of pathogenesis. She uses a broad range of human-relevant experimental approaches, which include in vitro and in vivo models, multi-omics and computational approaches, in vivo imaging, cognitive tests, etc., to explore pathogenic mechanisms and to identify novel targets and therapeutic approaches. I mean, there is a lot that one can say about Karen, but hopefully she'll tell us a little bit about her career trajectory to inspire us as well. So without wasting much time, Karen, the floor is all yours.
Thank you very much, Mahmoud, and thank you, Mosab, as well. I'm very honored to be invited to this seminar series and to get to know you better. I have been trying to find ways to sort of reach out more to colleagues across the globe, and to really sort of integrate our research and your research together so that we can go forward. My career trajectory is a little bit random. I started off working on cardiovascular developmental genes, and looking at mouse models with chromosome 21 abnormalities and looking at cardiac defects in Down syndrome. And that led me to a career in Alzheimer's disease through the chromosome 21 linkage to APP and Alzheimer's disease. So I didn't start with a mission of going forwards into Alzheimer's disease. I didn't always want to be a brain neuroscientist. So I reached it a little sort of circuitously. But I've progressed sort of fairly steadily from there, going through, looking at mouse models and where they've led me to doing MRI and therapeutics and genetics and biochemistry and cell biology, etc. So I think you'll see from my presentation that I haven't really followed a direct path, and that has been a great joy, actually, to be able to just sort of think big questions and apply them to the sort of problems of Alzheimer's disease. And that sort of takes me to something that I think is really important, is to look at the big questions, find the big questions that are remaining unanswered in the field of interest to you, and it could be any field, and really think about new and interesting state-of-the-art ways to actually answer a question. Don't get stuck in a rut with your Western blots and your immunohistochemistry, important though they may be. There are a lot of new methods out there to explore, apply, and you'll see that some of those methods, will feature in this talk. I have a personal connection to Africa, to South Africa. My family has lived in South Africa for a couple of generations, my father and my uncles, aunts, and etc. And my cousin Bridget might even be on this call. She's a doctor in Cape Town. And I really am very grateful to be invited to give this talk. So I shall get going. I'm going to be talking about a range of diseases called the tauopathies, and you'll see what that means in a minute. I'm just going to try and remove this banner that's right in the middle of my talk. Thank you. I hope you can see my talk. I hope you can see my cursor. So Alzheimer's disease, you probably had a couple of talks on Alzheimer's disease. I believe a couple of my colleagues have already talked before me. And the pathology of Alzheimer's disease really consists of at least two pathologies, which is the clumps of protein that form outside of the neurons in these amyloid plaques composed of this protein A beta. But inside the neurons develop these neurofibrillary tangles. And these are composed of an abnormal form of the protein tau. And they're going to be the subject of my talk going forwards. It's good to realize, though, that not only do these two proteins deposit in the brain of Alzheimer's disease, but also other proteins. And the two most common ones are alpha-synuclein and TDP43. So in something like 25 to 40% of cases of Alzheimer's disease, you have at least 4 pathologies developing. And I think this is relevant when you're thinking about therapeutic approaches to treat the disease that maybe target only, for example, A beta and the amyloid plaques, or only tau and tangles, is that you have these other pathologies almost certainly causing deficits in their own right. So in addition to the plaques and tangles, I'm not advancing. Why am I not advancing? Okay, there we go. In addition to the plaques and tangles, you have a significant inflammatory response, activated astrocytes and microglia. And although I won't be talking about these, the importance of the immune response, the inflammatory response, has really taken center stage now in the disease. And it seems to be coming in as both a driver of the disease and a driver of the progression of the disease, and interacting with all the pathologies and multiple pathways. And this is really something that has become a central part of the disease. In addition to the inflammatory and immune response, you have then the concomitant loss of synapses, synaptic abnormalities, degeneration and death of the neurons, cognitive impairment and death. So that, as we know it, is the pathology and the sort of the biology of the disease, and the progression, how the disease manifests to the neuropathologists, and really what we think is going on in the diseased brain. So one of the interesting things about Alzheimer's disease, and as you'll see with the other tauopathies, is that the two main pathologies, the plaques and the tangles, distribute themselves in different ways in the Alzheimer's disease brain. These 3 pictures show you 3 stages: prodromal, which means before cognitive symptoms manifest, the early to moderate cognitively impaired stage, and the moderate to late affected stage. And you'll see on the top the amyloid plaques and the tau, and the below tau tangles. And the distribution shown with these sort of heat map cartoons is clearly quite different, with neurofibrillary tangles starting in the entorhinal cortex, moving through the hippocampal formation and out into the neocortex, and amyloid plaques really being more distributed sort of more generally throughout the brain, focusing more on the frontal cortex, and really showing a density and a distribution that's fairly vague and diffuse compared to the very specific initiation pattern for tau tangles and its distribution through the brain as the disease progresses. Now, Alzheimer's disease is not the only degenerative disease that has tangle pathology, which we know as tauopathy, associated with it. You'll see here 3 different diseases: Alzheimer's disease on the top, progressive supranuclear palsy, and then Pick's disease. And PSP and Pick's disease are what's known as frontal temporal lobe dementia diseases. These are diseases which are also comprised of tau pathology in the brain, and they affect different regions. And I think looking at these different tau pathology associated diseases, we start to see something quite interesting, which is, although they all have tau as their primary, as one of the primary pathological proteins, the tau pathology starts in a different place in these different diseases, and it goes to different places as the disease worsens. And this is a very interesting observation, because how can a protein that is essentially the same in these diseases behave so differently? And this has been sort of one of the big questions that attracted me to looking at this problem, the problem of selective vulnerability, both in terms of initiation of the disease and trajectory to where the disease pathology spread. Now, the diversity of the tauopathies, the primary tauopathies and Alzheimer's disease, is not only manifest in terms of where the pathology is, but what it actually looks like. So you'll have neuronal involvement or glial involvement with the tau pathology. And here you've got on the top line, you've got AD and some of the primary tauopathies. And in the bottom, the second row of the glia, you've got the primary tauopathies, FTD, CBD, PSP, where glia are the only affected in those FTD diseases and not in Alzheimer's disease. The diseases differ by which type of isoform of tau protein is involved in the disease. The primary tauopathies, PSP, CBD, AGD, all have a 4-repeat tau protein associated with the disease. Alzheimer's disease has both 3-repeat and 4-repeat containing tau protein. The repeat aspect of that refers to the microtubule binding domain, and we'll come on to the biology of tau in a second. But this diversity extends not only in terms of the cell type affected and the isoform affected, but also the structure of the tau protein. Tau protein can take several different conformations. The fold, as shown here by cryo-EM, can be different between the different diseases. And there's something probably in this conformation of tau that probably impacts where the tau initially where it spreads to. And ultimately, that impacts the clinical manifestation of the disease. Do you get a deposition in the frontal cortex, which might impact your social and behavioral aspects, your behavior? Do you get it in the entorhinal cortex and hippocampus, which would give you a memory deficit? Do you get it in the brainstem, which would give you Parkinsonism? So these aspects of the biology of tau, maybe right down to the conformation, the structure of the tau isoforms, gives you a very high level impact in terms of the type of disease that you manifest. Now, the biology of tau, tau is a microtubule binding protein. Here you can see in the cartoon on the top, it's located mainly in the axons. It binds and stabilizes microtubules, and you can see the tau in the little orange flecks there. As a disease, a neurodegenerative disease, a tauopathy begins, the tau loses its traction with the microtubules. It falls off the microtubules in the axon, and it starts to accumulate in the cytoplasm in a somatodendritic compartment. It starts to be mislocalized. It's not in the axons anymore. The microtubules, as a consequence, destabilize, they unravel, and you ultimately get cell death. Now, what causes that to start? We don't know in a lot of cases. In Alzheimer's disease, we think there's the interaction with elevated A-beta, the amyloid plaque derived associated A-beta. But in some of the primary tauopathies, there are mutations in the tau gene that probably impact this the binding of the repeat domains in tau to the microtubules. How does the disease, what causes the disease to progress? We don't know. We know all of these stages shown on the right-hand panel the tau goes through those stages. We know it redistributes. We know it hyperphosphorylated. We know it changes conformation, etc. And we know it becomes so aggregated that it can't be cleared by normal clearance pathways, the ubiquitin proteasome clearance pathway and autophagy, and ultimately lead to cell dysfunction and cell death. Now, the order of those events is not quite known, other than redistribution into the soma and then cell death. But in between, we don't know exactly the linear pathway. So I'm going to come back to questions that came from the neuropathology studies, the post-mortem mapping studies. And the post-mortem mapping studies of Alzheimer's disease looked at patients who had died before they had a manifestation of Alzheimer's disease in their very early stages, middle stages, and late stages. That's what the mapping studies refer to. And one of the things that was noted by the neuropathologists, especially Heiko Brack and his partners, was that the pattern of tau pathology in the very earliest stages was very consistent. This stage on the left, the stage 1A, 1B, and 2, identifies tau pathology primarily in the entorhinal cortex. That little area with the red is the entorhinal cortex. And it starts in the transentorhinal cortex and it moves to the medial entorhinal cortex. It then moves into the hippocampus, and it then moves into the neocortex out in the other cortical fields. Excuse me. Now, the way that those fields are linked is neuroanatomical connections. There are neuroanatomical connections between those areas. And the fact that tau starts to appear in one area and spread very stereotypically through these other areas suggests that there's transynaptic spread of the protein or of the tauopathy through these areas and out. And that's the way that it spreads to other areas of the brain as the disease worsens. Now, we were interested in studying how this might occur. And to do so, we wanted to model tauopathy initiation and spread in a mouse model. And this series of pathology sections here stained with tau protein in brown show this in a real human brain, with tau pathology starting in the transentorhinal region, hippocampus, and out into the neocortex as the disease worsens. So to approach this, we use a transgenic mouse model, and here is a representation. We took a mouse that had the TTA transactivator protein under the control of a neuropsin promoter. Now, this drives expression of any transgene under its control in the entorhinal cortex. And you can see in black here, this is LAC-Z on the original neuropsin TTA mouse. The mouse we crossed it to was a tau mouse, the tau pathology. And this is the tau pathology you would see in a tau transgenic mouse. The first stage is when it's normally distributed in the axons. The brown stain is an abnormal tau antibody stain. And you move through the different stages with tau being seen in the cell bodies, accumulating as fibrils and becoming positive with dyes such as thioflavin S shown over on the right-hand panel. So when we made this mouse that had tau pathology directed to the entorhinal cortex predominantly, this is what we saw in a young mouse. The brown, again, is the tau antibody. And you'll see the tau is distributed here in the axons in the entorhinal cortex, more in the medial than the lateral. But you also see tau distributed here in the middle molecular layer of the dentate gyrus. This actually isn't tau expression. The only expression of tau is predominantly in the entorhinal cortex. This is actually tau that has flowed down from the down along the axonal tracts that terminate here from neurons originating in the entorhinal cortex. So this is just tau at the ends of the terminals. When we looked at an older mouse, and this was quite considerably older, about 24 months of age, we saw tau now accumulating in cell bodies in the entorhinal cortex, cell bodies in the granule cell layer of the dentate gyrus, cell bodies in the CA1 pyramidal layer of the hippocampus, and cell bodies in the extra hippocampal areas here out in the perirhinal cortex and out and beyond. And this is the pathology you would expect to see in a slightly later Braak stage human. This is getting up into Braak stage 2 and 3. And this is really starting to reveal that tau has shifted in position from here in the entorhinal cortex, now out into the hippocampus and out into the neocortical layers. One of the very interesting things about the design of this mice is that it allowed us to actually think about mechanisms. Why was tau that was originally distributed here in the middle layer of the molecular layer of the dentate gyrus now appearing in granule cell bodies? These granule cells are connected to the axon terminals there in the middle molecular layer by a single synapse. And here's a cartoon representing the monosynaptic connections. I keep losing my cursor, I don't know where it's gone. Here in the between the entorhinal cortex and the dentate gyrus. And when we were able to see in the old mice there on the right panel that tau are now accumulating in granule cell bodies where tau was not expressed, and that it had been in the terminals of the axons at an earlier stage, we were now able to say that tau molecules were able to actually cross across that synapse and jump into the dendrites from those granule cells that were coming, the dendrites coming from granule cells in the dentate gyrus. And this was really revolutionary because tau is not an extracellular protein, or at least it wasn't thought to be. It was a microtubule binding protein. And now we were seeing it hopping across a synapse from one cell to another. And we did some immuno-EM. Here is some immuno-EM, and you see the little spots of tau immunogold labeling. And you can see here the presynaptic compartment of MEC axon terminals and the postsynaptic compartment of granule cells, dendritic spines. And this area that we're looking at here is this area of the middle molecular layer. You can see it there on the left. Interestingly, we were able to see tau in the synaptic cleft, suggesting that it was crossing from one neuron to another. And we could see it in the presynaptic compartment, as you would expect, given that those neurons were originating in the entorhinal cortex. And you can clearly see it decorating what looked like synaptic vesicles. When you look at the mouse brains, the older mouse brains in 3D, as we did here using a technique that's a derivative of clarity, it's called iDisco. We labeled a cleared mouse, transgenic mouse brain with an fluorescently labeled antibody. And we were able to see the tau pathology distribution in 3D when we do these representations. This is a mouse hemibrain. We've taken off the olfactory bulb and the cerebellum, so it's only sort of part of it. But you can clearly see that the white spots of tau are distributed throughout the brain. And remember, these originated only in the entorhinal cortex. So now you're getting tau pathology spreading through the brain. This mouse, although it has predominant expression in the entorhinal cortex, it has very low level human expression, leaky expression coming from one of the transgenic mouse. And so what we think is happening here is tau molecules are spreading from one cell to another. And as they get into a new cell, a recipient cell, we think that the tau pathology recreates itself in a prion-like misfolding sort of process. And that then spreads from neuron to neuron throughout the brain using the human tau that's at very, very low levels throughout the brain as a template. And so you get this pathology spreading way beyond the area where it initially was seen. That bright spot that's just gone back round is the amygdala. And using our 2D sectioning immunohistochemistry studies, we had not gone into the amygdala, so we didn't know the tau was there. But the beauty of this sort of technique, and I would recommend it if you want to look at things in 3D and unbiasedly, is that you can start to see pathology in areas you didn't necessarily know it was going to be. So we've been very interested in mapping tau pathology in the brain, which areas are connected to which, what does that tell us about mechanisms by which it's spreading through the brain. But we've also been interested in looking at how the pathology spread correlates with a functional, a spread of dysfunction. Is this actually impactful at a functional level? Some of the studies we've done have involved functional MRI, but one of the studies we did, this one that I show here, is actually a study of atrophy using just MRI, not functional MRI, but MRI. And here we're looking, we've mapped out the atrophy shown in the blue and pink and overlaid it with the pathology we showed in the iDisco clearance pathology studies of the previous slide. And what you can see is that the atrophy overlays where the pathology is, but it actually precedes where the pathology is going to be. So the atrophy spreads into the hippocampus before the pathology does. And this suggests to us that we're not seeing cell body loss in those areas, but we're definitely seeing a loss of brain tissue, probably loss of synapses in those areas where the overt cell body pathology has yet to spread. So this is a very nice method, I think, for looking at, you know, unbiasedly. Again, you don't have to do cell counts, you don't have to do synapse count, synapse counts. You can now just see whether the volume of areas in the not stage, and you can look longitudinally because MRI is non-invasive and non-lethal. So one of the things we were also interested in was looking at the regions where this pathology developed in terms of its impact on cognition and memory. And the areas where the pathology develops in Alzheimer's disease are very interesting. These areas which involve the entorhinal cortex, the hippocampus, cerviculum, parietal cortex, are areas known to be involved in spatial navigation and spatial memory. And you'll see on the right there a list of the types of cells that are involved in these areas, the hippocampus and the entorhinal cortex. And you have place cells in the hippocampus, you have grid cells in the medial entorhinal cortex, and you have a lot of direction and border cells which are in these other areas that are also impacted by tau pathology in the very early stages of Alzheimer's disease. And what's interesting is that these areas are also affected in human, of course, which is why we're looking at this study, but that humans suffer from very early sort of loss of spatial navigation abilities. People get lost in familiar places. And we were wondering whether the impact of the pathology in these areas, and especially on some of the very important specialized cells, such as grid cells and place cells, might be underlying that pathology. I'm going to take 1 second break and go and get a glass of water if you just don't mind. Right, I'm back. One of the hazards of shouting at your computer on a Zoom all day. So we were very interested in looking at the functionality of the cells, especially the grid cells, because they've been reported to be dysfunctional in human Alzheimer's disease. So we did a study using in vivo electrophysiology, recording right in the region of the brain where these grid cells are located, which is the medial entorhinal cortex. And the grid cells are these specialized cells which are involved in spatial navigation. And as you are moving about in space, your grid cells are firing and telling you whereabouts you are in space. So you can imagine if you shut your eyes and you imagine going into your home, you have a mental picture of where your kitchen is relative to where you would stand. You can imagine yourself turning left or right to go to the kitchen or the bathroom, even if your eyes are shut. And this is because your grid cells and the other cells involved in navigation have made a mental image. So if these become destabilized, you could lose your orientation in space. What we saw when we measured cell functionality in our mouse models, and we used two different ages, 30 months of age and 14 months of age, ages at which pathology was either severe or fairly non-existent, we saw destabilization of these grid cells in this region of the brain in the mouse models, only at the 30-month of age stage, where the mouse had severe pathology in these grid cell fields. This was coupled with inability to navigate in mazes designed to test spatial navigation, especially the T-maze. And we saw some specific deficiencies in spatial navigation in these two mazes. We're now very interested in whether we can use the specific dysfunction in this area to devise better cognitive tests to identify abnormalities in cognitive abilities, especially in spatial navigation, at a very early stage in the disease, preferably before you have dementia as currently assessed in what is currently the prodromal phase. We're using a range of techniques, including virtual reality, along with in vivo recording, to actually identify whether there's dysfunction that can be visualized as a deficit in cognitive spatial memory, using games such as app-based games to test whether your memory is impaired non-invasively and without having to go into the clinic. And so there's a lot of work going on to actually identify what's going on in the brain regions of people very early in the disease before they show cognitive deficits, as assessed with our most used cognitive tests. So I wanted to switch gears a little bit now and talk about selective vulnerability. This is a very sort of fast-growing aspect of the field, and it's not just only Alzheimer's disease, but also all of the neurodegenerative diseases. because all of them show sort of regionality, and they show selective neuronal populations or glial populations that are impacted by the pathology. And they're different between the diseases, which is why you get the different manifestation disease. So it's become a really important question. Why? So for example, why does tauopathy only affect certain populations of neurons? Why does it show selective or differential vulnerability to the pathology developing in particular neuron populations? Now, the question of selective vulnerability for tauopathy has been studied and thought about for many decades. And the studies have identified cell type that is particularly vulnerable to tauopathy. But these studies have mainly been done either looking at the type of neurotransmitter that the vulnerable cell makes, their location in the brain, and perhaps their morphology. There's been very little work done to actually look at exactly the molecular signature of those vulnerable neurons. The reciprocal of vulnerable neurons is invulnerable neurons. And again, several pathways have been proposed to describe why some neurons are invulnerable to tau pathology. Now, we propose to use a computational approach to look at this question of why some neurons are vulnerable and invulnerable. And the first part of our study was to look at exactly what type of excitatory neurons are vulnerable. Sorry. So we started with an immunohistochemistry study. We used an antibody against tau, MC1, and we used antibodies against excitatory neuron markers, SATB2 and TBR1. And we used an antibody against an inhibitory neuron, parvalbumin. And we did a study in human AD brain in 2 regions at 2 Braak stages. And we were able to show that tau pathology completely co-localized with excitatory neurons and did not co-localize with inhibitory neurons. This was also the case in our EC tau mouse that I described earlier, that it co-localized with excitatory neurons. But there was also co-localization with excitatory neurons in human FTD. And FTD is, as I said, a primary tauopathy that does not have any amyloid. We've looked at 3 repeat and 4 repeat tauopathies, and again, excitatory neurons are always impacted. So what is it about excitatory neurons that makes them vulnerable? So studies, this lovely table here shows you the types of neurons that are impacted and the brain region impacted and what stage that they're impacted. And a number of different pathways come up as being potentially involved in why some of these neurons are impacted and others are not. One of them, proteostasis. There could be differences in proteostasis between vulnerable and invulnerable neurons, calcium homeostasis, firing rate. I can't see what's under my table here. Axon length and myelination, so morphology differences, endosomal lysosomal trafficking, cellular stress. There are various other pathways that we could look at, but we were interested in starting to look at what perhaps important in the biology of those neurons that impacted whether they would or wouldn't accumulate tau. So the first study we wanted to do was to look at proteostasis. And we were very interested in a study that had been done by Michele Vendrascola and his colleagues in Cambridge, which had identified various gene groups as being vulnerable in Alzheimer's disease. And the gene groups he put together fell into these clusters which were defining whether proteostasis could be active or inactive. And what he was able to do was he came up with gene clusters, gene groups, which defined a metastable proteome. And these are genes that are involved in keeping proteins soluble in a cell. So a lot of proteins are very at supersaturation levels, and which means that the slightest perturbation, it could be aging, it could be some beginning of disease, will tip them over the edge of solubility and they will aggregate in the cell. And a lot of genes that are involved in, associated with Alzheimer's disease control the saturation or the clearance of those supersaturated proteins. Other genes are involved in co-aggregation of proteins with plaques or tangles. They can promote aggregation, they can protect against it. And so various pathways involved in proteostasis came up as different between Alzheimer's disease and non-Alzheimer's disease transcripts from a transcriptomic study between different disease patients. Now, we were very interested in whether these genes that had been identified in these gene groups would differentiate between excitatory and inhibitory neurons. So did they go with vulnerable neurons or invulnerable neurons? We looked at two different data sets. Here, one from Naomi Habib and the Institute, and one from Kun Zhang and the Allen Brain Institute. And we were interested in what they had done here, because what they did was they were able to use PCA analysis to identify different neuronal populations. Here they show 8 excitatory neuronal populations and 8 inhibitory neuron populations. And they were able to see gene clusters that actually identified the type of neuronal population that they were looking at. When we combined the proteostasis genes with the excitatory and inhibitory neuron markers, we were able to show certain pathways went with excitatory neurons and certain pathways went with inhibitory neurons. And what we were doing here was really showing between these pathways, these proteostasis pathways, these genes involved in proteostasis that could sort of identify whether why excitatory neurons could be vulnerable and inhibitory neurons could be invulnerable. And this little table shows a subset of those. And for example, if you look at the protectors, BAG3 and HSPB8, they were more highly expressed in inhibitory neurons than excitatory neurons. And why that's interesting is because BAG3 is an autophagy related gene. So if it's more highly expressed in an inhibitory neuron, it's protective against the aggregation of proteins that would normally be cleared by that BAG3 pathway. What was also interesting was that a co-chaperone of BAG3, HSPB8, was also upregulated in inhibitory neurons versus excitatory neurons. So we thought we were on the right track here, that we'd identified genes that went together and that perhaps could be protecting inhibitory neurons. To get to the nub of why, of genes that were driving some of these effects, we did a hub analysis, and this sort of cuts through all the chaff and identifies the key drivers of some of these effects. And BAG3 was actually one of the drivers of aggregate protector pathways. So we thought that that was a good candidate perhaps for therapeutics, because it's the driver of protecting against aggregation, because it's inducing autophagy in the inhibitory neurons. So we've gone on and done some more studies now. We really want to get to this question of what is the biology of the neuron populations that makes them vulnerable or invulnerable. And we wanted to go beyond proteostasis. So we went back to the data sets, the RNA-seq data sets, which come from normal people. There's no disease in those people. And we pulled out gene clusters that went with the markers of our vulnerable or invulnerable neuron populations. You'll see TBR1 and SATB2 on the top, SST, parvalbumin, and calbindin on the bottom. And we pulled out other genes that really sort of segregated with those types of neurons. And you can see very nice segregation in the bottom between SST, parvalbumin, and calbindin gene clusters. And when we started to look at pathways that were differential between the 2 excitatory populations and the 3 inhibitory populations, we saw an interesting set of pathways involved in synaptic vesicle biology. So synaptic vesicle recycling, endocytosis, synaptic vesicle localization. You can see them circled at the bottom there. And we thought this was particularly interesting, that this might be a difference between, that it would explain why inhibitory neurons were invulnerable and excitatory neurons were vulnerable, might be because something to do with this that we saw, which is where tau molecules are actually stuck onto presynaptic vesicles at the point where transsynaptic spread occurs. So we thought this might be interesting, that perhaps something to do with synaptic vesicle exocytosis was carrying tau and allowing it to spread and accumulate then in other neurons, and that perhaps the vulnerable neurons, the excitatory neurons, had more of this going on, and that there was more firing, releasing the presynaptic vesicles that perhaps had the tau associated with them. This is hypothesis. We haven't tested this, but this is what the data is perhaps guiding us to think about. If you're interested in this sort of study, and I would very much recommend people do these bioinformatics studies, especially when you're in lockdown through COVID, it's a great way to entertain yourself. There are now a lot of data sets available. Here's just a couple of them, which are publicly available, and you can go and plow through and look for your favorite genes and identify some hypotheses and start to look at what genes go, with what, and start to really sort of use a computational approach to sort of create hypotheses. We tested that perhaps something to do with firing rate or firing was different between inhibitory and excitatory neurons by doing an optogenetics experiment. And here we were trying to increase the firing of a particular set of neurons, which were stimulated through an optogene being placed in the brain. We recorded with electrodes to make sure the firing was being stimulated by the blue light. And when we looked at increasing firing rate in a transgenic mouse, shown here, we could see that this optogenetically stimulated side had more tau pathology, shown in green, than the non-stimulated side. And we've done a number of experiments now, also using threads and using suppression of firing, and have looked at the impact of firing and brain activity on tau pathology, amyloid pathology, spread, etc. And one of our new papers is shown down at the bottom. Oops. It's not advancing. Okay. So I want to sort of finish off now and just come back to the idea about regional vulnerability, its importance in neurodegenerative diseases, regional and cellular vulnerability, and sort of bring you back to the idea that this is a common idea in all the neurodegenerative diseases. And this was a review that we wrote showing the various regions and the diseases impacted, or the diseases with the various regions impacted and the cell types that are impacted, showing that this sort of property of selective vulnerability is common to the diseases. And the review article sort of details some of the pathways that are thought to be impacted selectively. The other thing that's common to the diseases is the idea that the pathology starts in one place and spreads along predictable trajectories to other regions. You can see this for Alzheimer's and FTD, also for Parkinson's and ALS. So some of these ideas I think are common to these other diseases, and something really, I think, if you're interested in any of these diseases, this is a really fantastic part of the field to get involved in. Lots of good biology. You can use multiple different techniques. You can make hypotheses. You can create fishing expeditions and test your hypotheses. And you can look at mouse models, and ultimately you can look at human brains to make sure that you're on the right track. So this is a really great area to be thinking about, and very impactful in terms of predictions for disease progression and trajectory. Also for therapeutics, if you find a neuron type that has a specific makeup and is vulnerable to a particular protein, perhaps the therapy would be more impactful in that if you target it to that cell type. So lots of implications of this sort of these sort of approaches and analyses. And I just want to wrap up with a summary. We had shown that proteostasis or protein homeostasis mechanisms are important in determining the vulnerability of specific neurons to pathological tau accumulation. And this is really important, this one. Although the same mechanisms are likely to be involved in different diseases, neuronal subtype specific differences in pathway components and substrate levels determine selective vulnerability to different proteins. And this can give rise to different diseases. And what I mean here is that although we think of pathways and the genes involved in regulating autophagy or UPS as being common, they're not common to all neurons or glia. They have different interactors, different co-chaperones, as we saw with BAG3 and HSPB8. And they're present at different levels. They have different importance in different cell populations and different neuronal subtypes. I think there's something like 64 neuronal subtypes, excitatory neurons and inhibitory neurons. You know, there's now going to be hundreds of different neuronal subtypes, and they all make up that perhaps determines which substrate, and by that I mean tau, synuclein, Huntington, is vulnerable to what pathway deficit. And this might be very important when you're determining which drug to target, which type of protein in which cell type. A third aspect here, oops, lost my cursor again. Risk factors. Why do some things promote pathology? It maybe is that these risk factors impact particular cells. For example, aging is a risk factor for Alzheimer's disease, and it seems to set up deficits in proteostasis in excitatory neurons more than in inhibitory neurons. So perhaps just aging is a risk factor for you know, your excitatory cells being impacted more by tau pathology. Could genetic variants determine neuronal subtype and therefore clinical vulnerability? So we've been looking at gene variants, for example, for Parkinsonism in PSP. You have PSP, the disease has sort of two variants, one with Parkinsonism and one without. And we're wondering whether DNA variants impact certain cell types to either clear tau or not clear tau, and that would be in the brainstem. So if you have a variant, it might give you tau pathology in the brainstem, and that might give you the Parkinsonian version of PSP. So the disease manifestation, what you see in the clinic, might actually be impacted by DNA variants acting on particular neuronal subtypes. So it gets very complex, this interaction between factors that actually give you the clinical manifestation of a disease. Glia, we didn't talk about glia, but they have very high levels of protective genes, which might explain why most glia tend to be protected from inclusions, especially tau. But there is some interaction probably with the conformation of the tau. You remember at the beginning I was talking about the different conformations of tau, that they perhaps impact different cell types differently. And perhaps some of those conformations go into glia and accumulate in glia, whereas others don't. Hub genes, hub gene analysis is great for identifying, you know, pathways that might be more therapeutically effective. When you're looking at massive data sets of, you know, genes that are implicated from transcriptomic studies. So I would recommend hub analysis to sort of pare down what you might be wanting to look at for a therapeutic. And lastly, we really need far better knowledge of the genetic and molecular makeup of neuronal subpopulations than we currently have. There's been good initiatives. The NIH has an initiative to look at, the Brain Initiative to sort of really develop tools to look at neurons and to look at cells in the brain and pathways and circuits, etc. But we really need to understand the molecular and genetic makeup of individual cell populations, neuron populations, subpopulations, to be able to then identify why these differences are seen, why do different, you know, proteins accumulate in different neurons, what's the basis of that. So I want to finish there. I'm going to thank my lab, my new lab. I've moved to UCL in London. I have a computational PhD student, and I have a couple of people working on different aspects of tauopathy. We have my old lab at Columbia University in New York is still going, part of it. We have some projects ongoing there, and several people who've now grown up and out of my lab who led this work, especially Harry Fu, who's now at Ohio State University, Abid Husseini, who does all of the in vivo electrophysiology, and various others who've now gone on to bigger and better things. And I want to thank my collaborators, especially Michele Ventuscolo at Cambridge, and the people who have supplied the RNA-seq data sets, Broad and Allen, mostly ahead of publication, so that's great. And to thank all my collaborators, brain banks especially, who really underlie a lot of what we can do, and my funders, especially the UK DOI, who's currently funding the work, and the NIH, Tau Consortium, Bright Focus, and Servier Pharmaceuticals, who funded most of this work. Thank you.
Thank you very much. What a great talk. so much data and so much work within about 40 minutes. So thank you so much, Karen. So we've got about 10, 12 minutes for questions. But before I go to the questions, I'd like to tell all the audience that the talk would be on YouTube. And if you know or you want to give a talk as well, please do get in touch, because we want to make neuroscience accessible to all across the world. So yeah, so let's go into the questions now. And I'll probably start with myself. I was wondering, with the tau spread model that you talked about, have you looked at what, whether there is a specific species of tau that is responsible for the spread? Is it tau, oligomers, protofibrils, or fibrils, or is it a combination of all? And is post-translational modification also involved in that?
This is probably the hottest part of this hot field, is what exactly is seeding the tau spread and causing it. We've been, and the mechanism by which it's spreading, we've been taking a sort of unbiased, proteomics approach, but it's very difficult to narrow it down to exactly what is causing it, especially if you're looking at a mouse model where you've got overexpression, which is potentially an artifact, which might push tau into vesicles where it wouldn't normally be. So there's lots of theories, there's lots of hypotheses, there's lots of work on it, but it's just not really known how relevant that is to human always. That's the big question is, and the trouble with human is you're looking at sort of end stage disease. So you're not capturing that moment of spread. You're not capturing the sort of microenvironment where the tau is spreading. Very difficult to get at that, very difficult, but lots of theories and lots of data. Yes, phosphorylation has been looked at, other post-translational modifications, conformations of the tau, all sorts of things. So yeah, sorry, there's not going to be a definitive answer, I don't think, to that. Mechanisms, again, there's a couple of mechanisms for it spreading. One could be the neuron just degenerates and releases the tau and it just gets taken up by bulk in flow. Another could be vesicles, vesicles released. We like that idea. We've looked at exosomes and seen that these little tau seeds are carried by exosomes. And the other is by, maybe tubes like nanotubes, some other kind of connecting mechanism between neurons. But again, it's not known, and it's certainly not known in human brain, which is going to be the big problem, is how to work out whether any of what we see in the mice is relevant and real in human brain.
Absolutely, absolutely. So I'm going to jump into the Q&A, and in the interest of time, I'll probably just be taking those questions that are most relevant. I'm talking about post-translational modification. There is a question here: has self-acetylation of tau been observed in tauopathies other than AD? Could such behavior perhaps affect its distribution?
Again, acetylation is another, PTM that's being studied very actively. So, yes, it's been seen, its relevance relative to maybe glycosylation or phosphorylation, or, one of the other PTMs is unknown at this point. But yeah, it's certainly in there as a player.
Okay, fantastic. There's this question, I really like it. It said, do you think, well, let me start by saying, the person said many thanks for an exciting talk. So do you think the differential vulnerability to tau pathology between glutamagic and GABAergic neurons might also be related to the difference in their axonal length? If this is the case, might L5 pyramidal neurons be more vulnerable than L4 spiny stellate neurons, which make more local synaptic connections?
So axon length is really interesting, especially since we know that in the mouse models, if you overexpress tau in sort of long axon neurons, you get a lot of tauopathy. It's definitely one of the things we're interested in terms of morphology. It's very hard to know whether that's the cause of the vulnerability. You're sort of discussing about looking at these neurons in different layers is right spot on, because we're very interested in looking at vulnerable and invulnerable oddities. So if you've got a long axoned inhibitory neuron, is that invulnerable? Or if you've got this, the ones with multiple processes, chandelier neurons are inhibitory neurons, but they're actually vulnerable. to tau pathology, and they have sort of more spiky processes. They also have axo connections. So I think I can't answer your question, but what I would say is, if you know your brain anatomy and you know your brain populations, go and find the ones that, you know, where go and look at where tau pathology has been shown to be, and see if you can work out whether it is the axon length, you know, just by looking at the exceptions to the rule, or particular, you know, neuron populations that you know would fit the bill. So, I mean, I'm basically just sort of saying, yes, I agree, this could be the case, but you could, you know, if that's an interest to you, there's a lot of data on the neuron populations impacted, done by immunohistochemistry, and you can actually work out what it is that you think is, you know, relevant, axon length being one of those things.
Fantastic. So there is this question which said, as the different tauopathies have different configurations, could this be down to different mechanisms of initiation, or could it be due to solely because of different genetic factors in the patients?
Actually, no, I'd like to find that question because I lost track of it. Could it be, where is it?
It's in the Q&A. It's in the Q&A.
Yeah, I'm looking at the Q&A.
Yeah. If you just go to question from Abdullah Iqbal. It's actually the first question in the Q&A.
Okay, go up.
Yeah, because we've got like the comments as well as the Q&A. So questions from both sides.
Sorry, why don't you repeat it? I'm trying to find it.
So it's in the Q&A, the first question from Abdullah Iqbal. So it says, as the different tauopathies have different configurations, could this be down to different, have you found it?
No, but keep going.
Okay. So could this be down to different mechanisms of initiation, or could it be due solely because of different genetic factors in the patient?
Yeah, I mean, I think personally, I think it's the conformation. Now, why, you know, I think the conformation impacts what cell type it will initially, you know, deposit in, and then that might impact the trajectory because of the connections from that neuron population. You know, we know that the conformations of the tau are different in those different diseases, so it makes sense. Having said that, obviously, a number of things are different in those different diseases that could be impacting it. Personally, I think it's about conformation in, you know, being vulnerable, a particular conformation being vulnerable to a particular neurons, you know, makeup. But trying to study that is really quite difficult. It's very interesting question, but I don't know the answer to it, but I think it's great. It's a great question.
Yeah, so there's this question. So I know that we are probably have like 2 minutes. Do you mind adding like 1 or 2 minutes to?
No, absolutely. I've got all afternoon.
Oh, okay. So let's say we have 4 minutes. So yeah. So there's this question. It says, I would like to ask if you have looked or is interested in looking into glial cells in your model, as they are also involved in protein homeostasis, and nowadays it's more and more discussed the heterogeneity of these cells. Perhaps different populations of astrocytes have different roles supporting different populations of neurons, and it could be involved in the selective vulnerability. So I think this is more like a comment, and he wanted to hear your views.
No, I don't talk about glia at all because I have very little knowledge of it, but I absolutely appreciate it. It's important here. There's been some sort of data shown that if you take out glia, you don't get spread, and you know that the glia are perhaps the conduit through which these extracellular vesicles traffic seeds of tau between neurons. So it could be very important and different, you know, there's regionality to these glia. They have different sort of, you know, signatures depending on where they are in the brain. So lots of things could be contributing to the regionality, the initiation and the spread that goes through a glial component or an oligodendrocyte component.
Yeah, cool. So I'll probably pick 2 quick questions and we'll end it up with that. So quickly from my left, there is this question. It says the lack of tauopathy in inhibitory neurons does not necessarily mean that neurons are normal. So it's more like a question that is, it might be better to clear up intracellular tau, but there is still a lack of normal function.
Yeah, no, that is absolutely the case. And there's actually vulnerability to two things. One is to pathology development and one is to dysfunction. So you can imagine if you deafferent because your excited neurons have died, your inhibitory neurons are definitely not going to be normal, but they're not, so they might be vulnerable, in a different way. They're vulnerable to dysfunction. So absolutely, we just look at pathology because it's okay. It's easier for us to study. But we also, one of the reasons we do the in vivo electrophysiology is that we can actually pick up at least, dysfunction as measured by, firing rate, etc., to look at what's happening to other neurons in reaction to the pathology developing in one particular type. But yes, I agree. The selective vulnerability to response to pathology is also another aspect of this.
Yeah, absolutely. So I think I will ask this last question, and I have to apologize to all the people that couldn't read their questions. So this last question said, well, I like it because it's talking about, I would like to hear an expert, give a response about this. Can tau targeting treatments be effective? So this is more like an opinion. Yeah.
I believe so, but I think you're going to have to catch it early. So the thing with tau is that, I think it's sitting there in the medial temporal lobe early on. It's probably age-related increase, but it's not necessarily pathological. And it certainly doesn't cause much cognitive impairment because you don't see cognitive impairment until it's spread out of that area and into the neocortex. So I think if you can reduce the levels of tau either with an antisense oligomer, so you get rid of everything, or with clearance pathway, so you remove it, at that early stage when it's still prodromal, you can probably stop the rest of the cascade, the progression occurring, and it will be effective therapeutically. If you try and catch it late, it's already too late. The tau is propagated and is spreading through the brain. The thing to remember, though, with a lot of this is that you don't have to do much. You don't have to impact the course of the disease very much to have a big effect, because the disease, even if the disease slows down and is not stopped in its tracks, you'll still improve people's, life considerably. So I think tau targeting drugs will be more effective than the amyloid targeting drugs have been, at least but only if you catch them early enough in that prodromal phase. And that's why we're working quite hard to better identify that prodromal phase as being a tau, leading on to a tau-based disease versus some other vascular or something. You can imagine a prodromal phase. There's nothing, you don't know what's going to happen. So if you can improve the diagnostics because you're looking at the region, the entorhinal cortex, for example, hippocampal formation, which is the sort of area that, the area where this disease is going to start from, and identify dysfunction really, really early before you currently can with other tests, you might, and then administer your drugs, then I think you've got a much better chance. You can also help yourself with the tau PET imaging, which can identify tau in where it is. I think the region is more important than the density. And that's something you need tau PET imaging to tell you where that pathology is located. So, I mean, CSF markers and things are going to tell you probably the level. They won't tell you where it is. So I think PET imaging and amyloid imaging together with also other biomarkers is going to be very strong in telling you what stage of the disease you're at and whether you should give a tau therapeutic.
Cool. This is a comment. What advice would you have for ECRs? I mean, a lot of the audience would be ECRs like myself. And I mean, it would be nice to hear what your advice would be for ECRs to succeed in this.
Yeah, I mean, I think I spent a lot of my early years doing fairly standard techniques. I was a molecular biologist, although it was quite trendy in the 90s when I was doing it. And I think the best thing you can do to get yourself ahead is probably to learn a new technique. Learn a state-of-the-art technique if you're a postdoc, and have something that you can take with you. It helps you sell yourself in your next, when you want to move somewhere, you can be the expert in that technique. And, find a question that you can apply and use a really nice new technique. Go to the lab that's doing it best, go and travel, make some contacts, get yourself a little sabbatical money, you know, or travel money, and go and learn a new technique, apply it to a question. It usually gets you a better level paper, you know, and it gives you something that gives you marketability. So, you know, I think keeping your mind open to what new techniques, don't just rely on Western blots, immunohistochemistry, etc. You know, there are much better ways of doing things. I'm very interested also in the spatial. You know, we really need to get granularity on this. We can't just be taking bulk chunks of tissue and grinding them up and trying to work out what's going on. We really need to know at the individual sort of molecule, individual cell, you know, level, what's going on. And so any techniques that get you to that level, spatial transcriptomics, or, you know, single cell omics, etc., I think have got a lot of, you know, a lot of potential for the future. So my advice would be to make sure your skill set is state of the art, at least for one thing, you know, and then you start to think of big questions, and you can apply, you know, cutting edge techniques to a very big question. You know, the neuropathology, the selective vulnerability has been pondered for decades and decades. But, you know, how can you get real granularity to answer that, real specificity at the molecular level? And I think that really carries you forward. You know, that'll get you another job for sure. And, you know, from there you can go on. So I think keeping up to date. And, you know, when I go to SFN, or when I used to go to SFN, I didn't go to the talks on my disease. I'd go to all the interesting talks, you know, the big plenaries on a, you know, discovery of some technique or something, and think about how I could use that. And I think it's fun. You know, it's a bit of a long learning process, which is why I'd go to the lab that's an expert to get yourself in there, you know, get yourself a little travel grant, you know, and go and learn a technique and bring it back, you know, and then, you know, try and apply it yourself. So, you know, that's how I sort of started doing things. And I do that with all my postdocs. They all have something that they're going to learn, and they will then get a job, you know, because they've shown themselves to have sort of new skills, new ways of thinking about things.
That's absolutely very brilliant. Yeah, thank you very much, Karen, for a wonderful talk, as well as career advice. And to all those people that sent their questions, which we couldn't read, my sincere apologies. The time is so short for us to cover everything. And like I said, this talk will be on YouTube channel, so you can look it up. And if you want to give a talk, just do get in touch as well, please, and suggest other people. On behalf of the Society SONA Secretary General, Professor Amadi, I would like to thank you for giving this talk, and to all the audience for joining us today. Bye-bye for now.
Thank you.